Gay!
Hello, darkness, my old friend.
In the last two experiments, the alleles that will exhibit will be the same, or white and short, or pink and tall. But for example, if we try to combine two kinds of different flowers, like these ones, the offspring will exhibit alleles from the dominant alleles. For example, here the dominant alleles are the ones from this flower, tall and pink. So the offspring of these two flowers will be pink and tall. It will happen the same thing if we try to combine these kind of flowers. For example, if we try to combine the pollen of this flower with the pistil of this flower, their offspring will be the same, will be genetically equal. In another example, if we try to combine these two kinds of seeds, a green one and a yellow one, the yellow one, which have the dominant alleles, will show its alleles in the first generation, yellow and rounded. But in the second generation, the offspring will present characteristics from both of its parents, from the yellow one. Before explaining you the three principles of genetics, I have to show you the main terms of genetics. Gene, genotype, alleles, homozygous, heterozygous, phenotype, dominant, recessive, and finally the Punnett square. This one. Okay, now I'm gonna explain you my three laws. Law 1. The concept of dominance. The concept of dominance states that in the heterozygous condition, only the character with the dominant allele will appear in the progeny. For example, if we try to combine these two flowers, like in the example here, we will obtain an identical genetic offspring, like this one. If we try to combine a flower like this with a flower like this, we will have a flower like this. Why? Because these kind of flowers have dominant alleles. And in another example, if we are trying to combine two flowers like this one, as in the first example, our offspring will have the same genetical characteristics. Law 2. The law of segregation. The law of segregation states that the two alleles proceed by a diploid organism, segregate at the time of gametogenesis, and one allele goes into each gamete. For this law, I used to use these kind of squares, the punnet squares. For example, here we have the combination of a yellow plant with a green plant. The lowercase indicates a green, which is a recessive allele, and the capital letter Y indicates yellow, which is dominant. Here we can see that the offspring will be 50% heterozygous and 50% homozygous. Here is an example of the combination of a round and a wrinkled seed. The capital R are the round seeds and the lowercase the wrinkled seeds. Here we can see that the 100% will be heterozygous and that will be round. Law 3. The law of independent assortment. The law of independent assortment states that the list of different loci segregate independently of one another. Here we have another example where the one plant is tall and one plant is short. As we know, the tall allele is dominant and the short allele is recessive. So, in the Punnett square we can see that 25% will be homozygous for tall, 50% will be heterozygous also for tall, and 25% will be homozygous for short. For this law, I discovered two things, the incomplete dominance and the codominance. Incomplete dominance is a phenomenon where a dominant allele does not completely mask the expression of the recessive allele. The organism's phenotype may show the blending of both alleles. For example, flower color in Mittel's jalapa. It is also known as partial dominance or semi-dominance. This is an example of incomplete dominance. Both genes for red and white flowers are dominant. In crossing them, both are dominant and a pink flower comes out. The other one is the codominance. Codominance is a form of inheritance where both of the alleles are heterozygote fully expressed. Neither of the two alleles is dominant over the other. For example, the blood groups in human beings. As you can see, all the alleles are present in the final combination AO, AO, AB, AB. My thoughts. My only goal is to create an hybrid pea plant and observe the outcome. My time will come and my work will be appreciated. My interest in science is still growing. Now I'm attempting crossbreeding experiment with pot and bees.
In 1995, I performed a data analysis called the reproductive cells of the hybrids. And I can conclude that it was nearly impossible for Mendel to obtain such consistent data, even if he performed and made all the 5 experiments, interpolated and selected the best replication for each experiment.
In my point of view, I disagree with Mendel's method because he was using the wrong expected ratio in his testing of the 2/1 predicted segregation ratio for generation 3 families from generation 2 plants, possessing the dominant phenotype. I feel that this discrepancy was particularly strong evidence that Mendel misstated his results to better fit his model.
Everything changes.